Hello, my name is Teresa Trombetta, and I'm the Assistant Vice President for Alumni and Constituent Engagement here at Carnegie Mellon University, and also a proud alumna of Hines College. It's so great to see so many members of our Tartan community here today for this panel discussion on the CMU Mayo Clinic Transforming Transplant Initiative. I'd like to extend a special welcome to those attendees joining us from the Mayo Clinic. As you know, Carnegie Mellon has a tradition of bold collaboration, both within our community and beyond. We believe that when the best minds come together, like those that you'll hear from today from both CMU and the Mayo Clinic, there's no problem we can't solve or challenge we can't overcome. Organ transplantation is one of the greatest achievements in the history of medicine. And during this discussion, we'll hear from a panel of amazing faculty experts who will share their groundbreaking research that will help shape the future of this science. If you have questions, as I'm sure you will, as we hear from our presenters, please submit them through the Q&A feature at the bottom of your screen. We'll address as many as possible during the audience Q&A at the end. Now I'm so pleased to introduce Keith Cook, who will be kicking off today's session. Dr. Keith Cook is the David Edward Schramm Professor and Head of Biomedical Engineering at CMU. He also serves as the Director of the Bioengineered Organs Initiative and the Transforming Transplant Initiative. Dr. Cook's research focuses on the application of biomedical engineering to the treatment of lung disease. His primary research focus is the development of permanent replacement lungs for destination therapy. This work extends into many different research areas, including computational modeling and device design, development of novel biomaterials, and new drug approaches to reduce blood coagulation within those devices, and in vitro and in vivo biomaterials and artificial lung testing. His research also focuses on lung biofabrication, right ventricular function modeling, and pulmonary drug delivery using perfluorocarbon emulsions. Dr. Cook is a fellow of the American Institute for Medical and Biological Engineering. Keith, thank you so much for leading today's discussion. We truly can't wait to hear from you.
Thank you, Teresa. Good afternoon, everyone. I hope you and your families are well. We appreciate all of you joining us today as we discuss the Transforming Transplant Initiative. This initiative brings together two world-class institutions with the goal of making a real positive impact on organ transplantation. Today, our four outstanding panelists will share how their work in the fields of medicine and biomedical engineering and this groundbreaking partnership between CMU and Mayo Clinic has the potential to change the future of organ transplantation practice. Dr. C. Burchin Tanner is a professor of surgery and has been serving as the chair of the Transplant Center at Mayo Clinic, Florida since 2016. In January 2023, he was appointed as the Mayo Clinic Enterprise Chair for all solid organ and bone marrow transplant programs. Dr. LaTanya Hickson is a professor of medicine, the division chair of nephrology and hypertension, and the director of the Translational and Regenerative Nephrology Research Laboratory at Mayo Clinic, Florida. Her research mission is to advance clinical translation of promising novel therapeutics for diabetic kidney disease, and the most common cause of kidney failure in the United States. Her translational investigations examine mesenchymal, stromal, stem cell function, and cellular senescence abundance in diabetic kidney disease. Dr. Hickson brings research from the bench to the bedside, extending investigations from her research laboratory to investigator-initiated clinical trials testing the safety and efficacy of these novel therapies, including senolytics in individuals burdened by chronic kidney disease. Dr. Phil Campbell is a research professor in biomedical engineering, materials science and engineering, and biology. He has over 30 years of experience in multidisciplinary research, collaborating with basic life scientists, engineers, and clinicians across the areas of drug delivery, biomaterials, tissue biomaterial interfaces, biosensors, and regenerative medicine. His primary training is in physiology, with a focus on endocrinology, where he pursued growth factor regulation of tissue development toward engineering the stem cell microenvironment to spatially control angiogenesis, tissue regeneration, and the immune system. As you will hear today, his laboratory is also focused on extracellular vesicles, or EVs, including spatial control of their delivery, engineering surface and luminal contents of EVs for therapeutic delivery, including transplantation, and pursuing EVs as cell-to-cell communication modalities. Dr. Adam Feinberg is a professor in biomedical engineering and materials science and engineering at Carnegie Mellon University. His laboratory develops materials-based engineering strategies to control the self-organization and assembly of various cell types into tissues. Their goal is to understand the emergence of higher order function in biological systems, especially in ophthalmic, neuromuscular, and cardiovascular systems. His invention of the fresh 3D bioprinting technique enabled, for the first time, printing of complex 3D soft tissues at tissue and organ scales. He has been awarded the NSF Career Award and the NIH Director's New Innovator Award, and is a fellow of the American Institute for Medical and Biological Engineering, or AIMB. With that, I'm excited to turn things over to Dr. Tanner.
Thank you, Keith, for that introduction and for the opportunity to talk today. In the next few minutes, I would like to talk about how physicians and scientists at Mayo Clinic are partnering with scientists at Carnegie Mellon University to find solutions and to create a better future for patients in organ failure and need organ transplantation. Many of you in the audience may not be familiar with organ transplantation. Considering the long history of medicine, organ transplantation is a very new medical field. With relatively rapid development of this field over the last 4 decades, over 1 million organ transplants have been performed in the United States. While we celebrate the successes of organ transplantation, there's the other side of the picture that causes unmet patient needs. There's increasing number of patients with organ failure. Transplant still depends on organ donors. However, there's limited number of organs available for transplant. Therefore, the number of patients who need organ transplants paces those who donate organs. In fact, most patients who wait for an organ transplant today will never receive an organ transplant, and approximately 20 people die each day in this country waiting for an organ transplant. It's a tragedy for patients, for their families, and for society to see patients die. For us in the field of organ transplantation, we witness this tragedy every single day. There are major challenges. While transplant depends on organ donors, not every organ that is donated can be transplanted due to concerns of organ quality. It is never certain whether a patient who needs a transplant will definitely get a transplant. We're never able to tell our patients when exactly they will be able to receive an organ. And after transplant, the outcome still depends on organ anti-rejection medications, which all have major side effects. Organs that are transplanted may not last a long time, and some patients may need repeat organ transplants. Patient care waiting for an organ transplant, transplant process itself, and care after transplant can be very costly to the patients, their families, and to society in general. The ultimate goal for us is that no patient should die waiting for a life-saving organ transplant. To be successful in our quest, there are 2 major categories of problems that we need to solve. On one hand, we need to address the increasing number of patients with organ failure. On the other hand, we need to address not having enough organs for transplant. Systematically approaching in each of these two categories, there are two potential solutions. We're aiming to prevent organ failure in patients so that in some patients we can perhaps prevent organ failure from happening. And also in patients who already received organ transplants, we're aiming to prevent organ rejection so that we decrease the need for second transplants. On the other hand, we are aiming to restore donated organs that are inferior quality so we can increase utilization of those organs. And of course, using technology, we are aiming to engineer new organs that can be transplanted. There are four goals of Transforming Transplant Initiative. We aim to restore donated organs so that we can optimize organs for best outcomes and decrease organ discards. We aim to prevent organ failure in patients who have received transplants before. Our idea is that one organ for one patient and significantly decrease repeat transplants by prolonging life of a transplanted organ. We aim to come up with solutions for early diagnosis of organ dysfunction so that we can hopefully prevent the need for organ transplants for many patients. We aim to develop technology to engineer new organs so that when an organ transplant is needed, we eliminate all of the uncertainties, we eliminate the wait time for transplant, and an organ can be custom made for the patient and transplanted at the optimal time. We are imagining a future where we decrease our dependence on organ donation, where we can refurbish less than optimal organs. By custom designing organs, we can liberate patients from anti-rejection medications. By having more available organs, ideally every patient who truly needs an organ can get an organ at the optimum time with decreased cost to society, and those organs last long lives. As we embarked on the journey of transforming transplant field, Mayo Clinic is partnering with Carnegie Mellon University. Mayo Clinic has nationally recognized transplant programs. We have been providing consistently high quality of care for patients in the last 60 years. We have large transplant programs in our 3 campuses, and altogether, Mayo Clinic is now the largest organ transplant provider in the United States. Each of our campuses has established innovative regenerative medicine programs which collaborate with the transplant programs. And many of you know well that the strengths of Carnegie Mellon University, its very strong engineering programs, strong focus on tissue and organ engineering, 3D biofabrication, computational organ modeling, biomaterials within the biomedical engineering department. With this initiative, by working together, we are bringing top medical and engineering expertise that ultimately will affect millions of lives. Instead of waiting passively for the future, we are taking an active approach to create the future. Next, you will hear from 3 of our investigators working to solve these challenges, and I will pass the screen to Dr. Tanya Hickson. Thank you.
Thank you, Dr. Tanner. I'm excited to talk about what I consider to be one of the most important organs in the body, and that includes the kidney system. Chronic kidney disease affects 1 in 7 adults here in the United States. And as you may imagine, the most common causes, and Dr. Cook mentioned this earlier, common causes of kidney failure are diabetes mellitus, as well as high blood pressure. But there are certain groups that are at risk for kidney disease. In particular, those are individuals that are of racial and ethnic minorities. And how about those of us that are thinking about retiring, that good old age of 65? We think about what we're going to do with all the resources we've invested over time and the time we'll spend with our loved ones. But kidney disease is also a disease of aging. And therefore, we find that in our patient populations, there are many more mature individuals that experience chronic kidney disease. I imagine today, as you drink your coffee and your water, you found going to the restroom an annoyance. But I'd like to tell you that there are over 800,000 individuals in the United States that only wish that they could go to the restroom and have their kidneys filter their bloodstream, get rid of those waste products and the extra fluid that their body doesn't need. As you can see, this has increased over time. And unfortunately, we still have 88,000 individuals that are waiting for a kidney transplant. Now, while we celebrated transplanting 25,000 individuals with a kidney for the first time in 2022, that left us with over 60,000 individuals that were waiting on that phone call, telling them that we had a kidney ready for them. Our research is regenerative cell-based therapy research. And how are we applying this in these bioengineered organ initiatives that we're doing with Carnegie Mellon? We are hoping to improve both the kidneys that we were born with, as well as the transplant kidney health across the disease timeline. So when we think about this, we want to delay kidney disease progression prior to transplant. And then after transplant, we want to delay disease progression, and we want to limit rejection so that we don't have to have a second organ that's transplanted. In this particular initiative, we're also hoping around the transplant timeframe to enhance organ suitability for transplant. With this, we are implementing mesenchymal stromal cell-based therapy and their extracellular vesicles. And as you can see in the background, we have these beautiful fibroblast-looking cells, and they release cytokines, growth factors, and extracellular vesicles that can help repair the kidney by reducing inflammation, stress to the kidney, and scarring. The wonderful thing about these cell-based therapeutics is while we like the work that they can do in the kidney, which you see over on the right-hand side, they also have effects outside of the kidney, where we can preserve islet function in the pancreas, reduce adipose tissue inflammation, enhance insulin sensitivity, and not pictured here, but also delay or improve the response that we need in order to preserve that kidney allograft. We're designing cell-based therapies for clinical trials. And when we think about this, we're looking at individuals with kidney disease. We can either take the patient with kidney disease, or we can take a donor, a healthy donor, and provide allogeneic stem cells. We process them, cryopreserve them, and give the stem cells to the patients and turn that diseased kidney into a treated kidney. I'm excited to share that here at Mayo Clinic, we believe are the first and only center in the country that's delivering mesenchymal stromal cell therapy in clinical trials. We're doing this for diabetic kidney disease and chronic kidney disease. And I cannot tell you how many patients email us on a daily basis asking to participate in the trial or find ways that they can receive these stem cells here in the US. But we're focusing today on what are we doing around the time of transplant. And as this relates to cell-based therapy, I'd like to give you just a little bit of background. Most of our transplant kidneys come from donors after their death. Now, many of you may know someone who's graciously donated a kidney to a loved one, or maybe even anonymously, but that happens in 3 of the 10 kidney transplants that are performed. Therefore, the vast majority of transplants that go into patients' bodies for the kidney are from deceased donors. If we're hoping to improve the rates of transplantation and eliminate that 60,000 people that were left there without a kidney, we need to reduce the number of discarded kidneys. It's imperative. When we look at these numbers, 21 to 25% of kidneys that our surgeons go in across the country and harvest the kidneys from those people that donated a kidney after death, we are discarding 1 in 4, so 1 in 5 kidneys. Transforming transplant, our mission is to reduce that number or that percentage of kidneys that are donated or discarded to increase the number that can now be used for transplant. And we're applying cell-derived therapy for this initiative. We came together as a group with a nephrologist or a kidney specialist, myself, a transplant surgeon, Dr. Shannon Mao, and our beloved Phil Campbell, Dr. Campbell, our research professor in biomedical engineering at CMU. And our project is focused on extracellular vesicle-based therapeutics for ex vivo preservation. So taking that kidney that's been harvested, optimize the kidney such that we can salvage a kidney that may have been discarded, and therefore increase the number of individuals that are able to receive a kidney transplant. With that, I'd like to turn it over to our expert for extracellular vesicles, Dr. Phil Campbell. Thank you.
Hello, everyone, and I appreciate that introduction. And I'm going to focus on extracellular vesicles and explain what they are, because it may not be familiar to some of the audience. And basically, they are defined as these vesicles which are contained with a plasma membrane, much like your cell membrane. They range in size from over 1,000 nanometers down to less than 50 nanometers. We usually work in the range of 100 nanometers, so they're true nanoparticles. Now, every living cell in your body makes them, even the dying ones, but not just in humans. Every living cellular-based organism on Earth makes extracellular vesicles, or EVs. So that means from bacteria to plants to you. Now, I refer to EVs as universal communicators. Why? Because they carry various cell communicating cargo, from steroid to protein hormones, to peptides, to proteins, to DNA, RNA, even cellular organelles such as mitochondria. So therefore, they can be potential therapeutics. EV cargo also reflects their cell source. So they are potential biomarkers such that they could be markers for cancer or various disease states. Now, the cargo can either be on the surface or within the lumen. Now, just like mythological Janus, EVs have two sides. Over the last 20 years, there is evolving evidence to suggest that EVs direct all normal physiological processes. But on the flip side, there's evidence that also suggests that EVs can direct disease initiation and progression. Now, as we know, chronic inflammation leads to a disease progression, to a disease state. Ultimately, that the final solution is transplantation. Now, this disease progression event involves EVs. On the flip side, what we're trying to do is pursue EVs as a mechanism to disrupt this normal disease progression, limiting disease development, or even reversing this disease phenotype to a more normal phenotype. Should transplant be necessary, these EVs can hold, we are looking at them as potential therapeutics to improve transplant outcomes. Now, as Tanya mentioned, we're working with adult mesenchymal stem cells. And so what we are looking at are adipose tissue, bone marrow, and most of us have excess of adipose tissue around. And also we can isolate them from differentiated fibroblasts. We can induce them to pluripotent stem cells. But in any case, the cargo that these EVs carries reflects that of their cell source MSCs. Now, why we're using them is they can potentially simplify some of this mesenchymal stem cell therapies because they're cell-free, so the logistics can be a little bit different. However, it isn't that easy. There are challenges to developing these therapeutic applications. There's issues with stability, rapid plasma clearance if they're injected in the blood, also delivered in the blood, targeting issues, how to get it to the specific organ you want it to go to. Balancing functional outcomes, which are typically measured with understanding the mechanism of action of EVs. There's also manufacturing, scalability, and lot-to-lot quality control issues. And at least when we got started in this area, there were limited tools for functionalization. So how my lab is addressing this is to engineer the EVs both on the inside and the outside. So we're developing strategies to load the luminal space of these EVs with things from proteins to nucleotides. We're also developing strategies to load them with hydrophobic drugs and small molecules. Once loaded in the intraluminal space, they're protected against deactivation in this external environment. Also, the EVs helps to better target them to their desired cell types that we want to address. Also, we are modifying or engineering the cell surface using a platform approach that's based on cholesterol DNA anchors, as well as click chemistry approaches to immobilize on the surface small molecules, proteins, and even polymers. All of these with the intent to help to rectify and simplify better targeting, better stability, etc. Now, along with the CMU Mayo partnership, what we're doing is trying to use EVs as a part of these practical applications, and ranging from postponing transplantation by hopefully postponing the chronic development of organ disease, as well as improving transplant outcomes. Now, these will be involving both systemic delivery strategies as well as local delivery strategies. And Adam will be talking about bioprinting. So these EVs could actually be incorporated in that approach. And as Tanya was talking about, we're also interested in developing EVs for organ perfusion techniques, because this is delivering these EVs in a unique environment where it addresses a lot of the shortcomings of systemic delivery. And lastly, our focus is on both kidney, liver, and the lung. Now, transplantation and the promise of EVs. Where are we at? Well, understanding the EV basic biology remains a critical component to developing EV therapeutics for transplantation. There are many challenges in transplanting EVs, but we believe that they can be addressed using engineering approaches. And lastly, although we've known about EVs for 60 years, their potential as therapeutics is less than 20 years old. So our journey is just beginning. And with that, I will pass on to Adam.
Great. So thank you very much, Phil. And really excited to kind of share with you the work we're doing here at Carnegie Mellon that really is synergistic with the efforts of Mayo Clinic to really, look to solving the organ transplant problem by actually rebuilding organs from scratch. Very much a moonshot in terms of, kind of biomedical and bioengineering technology, but one that really has the potential to broadly solve the lack of enough transplant quality organs for the number of patients who ultimately need them. So, I just want to kind of quickly cover what bioprinting actually is. And so really what bioprinting is the adaptation of 3D printing technology that has really excelled over the past, say, 20 years for using metals and plastics and ceramics, and really transitioning that technology to printing with living cells and other aspects of tissue. To do that, there are a lot of unique challenges that we have to work on the hardware side, including printing under sterile conditions with living cells under conditions that are within a really limited range of temperature and other conditions that are compatible with living systems. With that, we have to develop novel and new bioinks and biomaterials. These are essentially what the printer is actually printing with, and this includes living cells and things like collagen that make up the main parts of our tissue. And then technologies that allow us to actually generate enough cells to actually have the quantity we need to print a tissue, which is actually billions and billions of cells for something like a standard organ. We need to be able to vascularize that organ, so basically provide a blood supply to every single cell within the tissue we're building. This is something that our body does incredibly well, but is really hard to do when we're trying to build a tissue. And then we also want to build in the immune tolerance so that we could hopefully eliminate the need for anti-rejection medication by using cells that are intrinsically compatible with a much wider range of people, so we can limit the drugs that really help transplant be viable, but also have a number of adverse side effects. And then finally, it's really the validation of both the structure of what we're printing and then ultimately its organ function. And I think it's important to keep in mind that we can build organs that look like the organs that we're replacing, but they don't necessarily have to. And so we're exploring a much broader design space of organ replacements that would be able to restore function. And it may look different than your existing organ, but in terms of efficacy are just as good. And so how do we get to organs? And so, you know, this really is a moonshot, a very kind of big reach, if you will, for where we are today. But we've really been trying to chip away at the key aspects that we need to get to organ replacement. So what are some of those? If we, for example, want to build a heart, well, we obviously need to build things that are big, right? So as big as real organs. And that might sound obvious, but you know, bioprinting to date has largely focused on building tiny little structures that we would evaluate in a petri dish. How do we move that to large scale tissues that are really realistic and relevant for therapeutic use? How do we achieve the right kinds of physical properties? Think about a heart valve that needs the right kind of mechanical strength so that it can flex billions of times over someone's lifetime. How do we recreate that multi-scale vasculature, right? This is everything from large blood vessels like arteries that might be over an inch in diameter, all the way down to capillaries that are only about 5 to 10 microns in diameter. That's about 1/10 of the diameter of a human hair. And then how do we print with living cells at high density so that we build tissues that have the right level of function that we need? A technology that we've developed in my lab at Carnegie Mellon and has been kind of adopted more broadly across the biomedical community is one that we call FRESH. It's an advanced biofabrication platform. And if you look at the cartoon in the middle, you can see that what this really involves is the ability to take say a needle and a syringe that can extrude materials and then print them inside of a 3D environment. Now with that, we've been able to do some very exciting work. So we've been able to take collagen, which is the main structural component of the human body, along with heart muscle cells, and print them into initial models of the human heart. So this is like a small model of the left ventricle. It's about the size of the end of your pinky. We can control the alignment of the cells. And then when we print that, we can see that under the microscope, when we are watching the active contraction, we can build these little ventricles that are highly contractile. They conduct an action potential, and we can synchronize their contraction. And then when we look at them from the top down into the ventricle that we printed, you'll be able to see kind of that wave of activity propagating. And then when we synchronize contraction, we get wall thickening and some decrease in chamber volume, which is basically what happens when you eject blood out of the heart. So really exciting first steps in building heart muscle. We can also build heart valves. Here we see a heart valve made entirely out of collagen that mimics the structure of the real heart. We've shown that can replicate the function that we need the heart valve to do in terms of the pressure and flow rates and seen in the real body. And we can also extend that to building the whole heart as well based off of medical imaging data. And then another really interesting aspect then is taking that forward towards translational applications. And so here you see an example of building a scaffold for a loss of skeletal muscle. We have a patient here who had a traumatic injury to one of their quadriceps, and we can actually mirror the muscle from the good side to replicate what is missing from the defective side and create these scaffolds. And these scaffolds have intrinsic architecture, as you can see in these images here, to help guide the formation of muscle, and not just parallel muscle, but all the more complex structures we see in skeletal muscle throughout the body to restore function, which is relative to skeletal muscle as well as the heart. And now we can think about taking these technologies and using them to start to build organs. Now, so the organ, while we would like today to build something like the whole heart, we have to recognize that that's still a little bit beyond our technological reach. However, we can start to think about development where the heart starts as a heart tube, and then it becomes the 4-chambered heart we all know during embryonic development. And we don't copy embryonic development exactly, but we try to be inspired by it, where we can look at what changes in the heart during development and the changes in matrix, the loading in the heart, and ultimately to start to build organs. And in this case, when I say we're building organs, we're trying to build an organ that is representative of early in development when the heart is much simpler. So in this case, a heart tube. And so here you can see that we can print these heart tubes. Here you see the cardiomyocytes. In this video here, you can see they're electrically active. They're actually contractile. And then they can actually pump beads. And so this is really our first foray into building functional organs, starting with a simpler organ early on in development. And as our technology improves over time, we'll continue to build more and more sophisticated organs that generate, in this case, like the heart, higher pressures that become more realistic for transplant as we move down the line. And so I just want to close with this last slide, projecting the timeline of some of these technologies to the clinic, because I think this is always important to think about where we are today, where are we going, and how long will it take to get there. And this shows about 20 years. It will take about 20 years, I think, to go from where we are today, which is in 2022, we had the first 3D bioprinted example of a tissue implanted into a human patient under an FDA clinical trial. This was for actually an ear, so really for more of a cosmetic application. And while there's a number of steps on this timeline to get there, I really do think in about 20 years, we'll be at the point where you'll see 3D bioprinted tissues and organs as viable treatments for transplantation in patients. So with that, I'd like to welcome back the rest of the speakers for today and move on to the panel discussion.
Thanks a lot, Adam. I appreciate that. Everyone, we are going to get into the question and answer session. And so I want to thank everyone who has already provided some questions in the Q&A. And so we will start answering those, but I want to encourage everyone else to be brave and throw some more questions in the chat. We'll be happy to answer those. So I wanted to start with one of my own questions, and that's, I noticed that several of you were mentioning using stem cells in your research. So how do you generate the stem cells that are used for organ repair and production?
I can get started, and then I'll maybe Phil, you can tag behind me when we talk about what comes from the stem cells a bit. Well, as Dr. Campbell mentioned earlier, many of us probably are able to give up a little bit of adipose tissue, particularly here in the United States. But we tend to, at least at Mayo Clinic, from our biomanufacturing perspective, our focus has really been on adipose tissue, which is highly abundant, and many people are willing to donate this. But as I showed in my slides, we can utilize bone marrow, right? You can even get it from dental pulp. And there are a variety of tissue sources for these mesenchymal or mesenchymal stem and stromal cells that we're using in these therapeutic applications. So one, we're getting it from consenting adults. Two, we're trying to decide if we should utilize tissues from individuals that have disease or get it from a healthy donor. But for the most part, these are the sources that we're using, and have found some therapeutic benefits in kidney disease and other organ systems.
But and I'll follow through and just say that, like Tanya said, it also comes down to having enough cells to be therapeutically relevant, whether you're going to use those cells in themselves, like Adam was talking about, for printing purposes, or maybe we want to use them as a source of EV. So we need to pick a cell source, adult mesenchymal stem cell source that's readily available, such as bone marrow or adipose tissue, though most tissues in our body contain resident stem cell populations. But alternatively, too, is we can induce those cells as well. So genetically induce them to be mesenchymal stem cells. Could potentially allow us to make stem cells that represent each of our specific patients. So, and then those would be a permanent cell types as well. So those are also the advantage.
Yeah, and I can just add that, in a lot of the work that we're doing, we are using something called an induced pluripotent stem cell, which is where you would take a skin cell from an adult, and there are some methodologies developed where you can essentially reprogram that adult skin cell into an induced pluripotent stem cell, which you can then kind of take and make to almost any cell in the body, whether it's a cardiomyocyte or a heart muscle cell, like I used in some of the examples I showed, or a mesenchymal stem cell, similar to what Phil and Latanya were referring to.
Thanks a lot, everyone. So this is another question I think that is good for all of you, and that's, what is the current status of FDA approval for biologics, such as extracellular vesicles and bioprinted organs? And when do you think we might see, I know, Adam, you touched upon this, but when might we see such products on the market?
I can start off here as well. Adam, that was really perfect. We got a timeline question right when you were doing a timeline slide. It was the best cue right there. But at this point, when we think about applications, let's say, for example, in my world, where I'm treating patients with diabetes and chronic kidney disease in hopes that we can delay the rate of progression of kidney failure. Right now, there are no FDA approved in the US applications for mesenchymal stem cells. However, they are approved in Canada and Japan, and I believe China. And this is primarily for the indication of pediatric refractory graft versus host disease. What we'd like to do is see this progress, but I imagine that the time frame at least is 10 years. And where we will have to move from these phase 1 clinical trials, which I'll add are pretty expensive to do. We need to first demonstrate that their safety, which we've seen in multiple trials, that these cells are safe in their therapeutic application in healthy and diseased individuals. And then we want to move on to the phase 2 and phase 3 trials that are necessary to move through that approval process with the FDA. So I'd say a minimum of 10 years. I wish it were more, but I do want to talk about medical tourism and try to dissuade people from leaving the United States, moving away from FDA assisted or allowed studies to go get these cell-based therapeutics in other places where there may not be the oversight that's needed to keep people safe with these new regenerative therapies.
Thank
And
you.
I'll mention one word about EVs. Oh, sorry. So they're well over 200 clinical trials ongoing worldwide right now, from phase 1 to phase 3, but none approved at the present time. But I would think that some of them should be approved in the next 5 years. So that's a reasonable projection.
So I think that moves nicely into one of the questions that we got. So Phil, I know you talked about, EVs coming from all cells, including those that have disease. And so I'm wondering if you could comment about differences in EVs that are coming, for example, from cancer cells and those that we plan on using for therapeutic purposes.
Right. So since the EVs reflect their cell source, I mean, the cancer cells are going to carry all of the information that is basically associated with their parent cancer cell. Not only will they reflect, be used for biomarkers, and you can harvest these out of blood, and then you can predict whether that person has cancer or how they're performing in cancer treatments. They also carry the ability to not necessarily create a new cancer cell, but to transport some of those cancer phenotype to cells they go to. So we do not want to use disease cell EVs for our therapeutic potentials. That's why we want to go with the stem cell based stem cells, because they're not going to have that. They're not going to carry that type of information.
Thank you. So Adam, here's a question that's very near and dear to my heart. So is the design principle form follows function applicable in bioprinting artificial organs, or are there cases where amorphous shapes are better alternatives for various reasons?
Yeah, I mean, I think I touched on this maybe ever so briefly when I spoke, but our organs look like the way they do for a number of reasons. One is that they've evolved for a high level of functionality, obviously, but we all started out as a single fertilized egg and had to become a full human being. And so the evolution of the organ also had to be able to kind of fit within the confines of that process. So when we think about creating a replacement organ, obviously we can recreate the existing organ. We can take an MRI or CT image and use that as our design input and create something that matches that architecture. But we also have the freedom to think more broadly and more creatively, right? So if we think about the heart, which needs to pump blood, does it have to look exactly the same way as it does in the body? Not necessarily. And then, I think as you were referring to, Keith, when we think about the lung, and if those who aren't familiar, the lung has this kind of complex branching structure, almost like grapes, like a thing of grapes. And that's great, but are there alternative, designs that'll still allow excellent gas exchange? And I know, Keith, that's one of the areas you work in quite heavily. And so I think really we have an opportunity, right, that we can kind of use the existing organ as a starting point, but also have the flexibility and the creativity to think more broadly to hopefully come up with a solution that perhaps is just a lot easier and less expensive to manufacture, for example. Even if that was all we could achieve with a different design, we would still serve a much larger patient population by doing so.
Thank you. I'm going to direct this one to you, Burchin. And so we talked about a lot of different technologies that'll be brought to bear here as part of the Transforming Transplant, a lot of different areas that we're going to be working in. What are the gaps that need to be closed that you're most excited about, Burchin?
There are many gaps. So if you remember those 4 different areas that I mentioned that we're focusing on, if you take one of those as the organs that are donated, but we're not able to use them essentially. So that's a big gap actually. So American population is very generous. They donate organs and we recover those organs. Unfortunately, we find that the organ quality is low actually. And with this current technology that we have, we feel that we cannot transplant that organ safely and promise to the patient that we will have a safe, great outcome. So, an example will be livers that are donated, and we find that there is the fat content of the liver, donated liver, is very high, and when we transplant, it's not going to work. So, what if we take that organ and we place on a machine, and then with time, over the next several days, we could decrease the fat content of that liver and make it usable. So this is one of the areas that we're very interested in, making organs better outside of the body. And that's a big gap with the current clinical practice. It's a big gap in technology. But this could be one of the low-hanging fruits that we are focusing on right now.
Thank you very much. So I appreciate all of the questions from everyone. We are nearing the end of our session, and I have a burning question that I want to ask all of the panelists. So I apologize if we didn't get to your questions. So my last question for everyone is, what are you most excited about related to this partnership? And I'll let you jockey for who's going to answer that question first.
I'll start. So this has been a great collaboration between the 2 institutions, and then this is not new. This goes back 3 years, and we've been able to develop this relationship during the pandemic, actually, over video calls, and then we built we understood our cultures, and we built this relationship. So that tells you how we sort of clicked, even though we were geographically separated from each other. So that tells you the future of this, collaboration. That's one thing. The second thing is this is not just a research, but this is really a research innovation project that's really going to bring technology to patient care in an accelerated manner. I'm very excited about that. And we're essentially bringing top clinical expertise and top engineering expertise. We start understanding the problems because we look at the problems in different angles, actually. So that's the second one. And the third one is as important as the previous two is we're investigating in terms of developing educational programs to train future generation of tissue engineers, biomedical engineers, clinicians that will understand the biology and engineering at the same time. And I think I'm excited about that as the other, research part of this. So that's my opinion.
Yeah, maybe I can jump in and just say, I think, one of the exciting things about this collaboration is I think we all realize that we can do better and that we need to do better than what we've been able to accomplish to date, which is still, I think, quite impressive. But, there's all this compelling need. A lot of what we're trying to do as well, we all believe it is possible, right? Not if it's possible, it is possible. These improvements, these innovations are going to happen, but it really requires collaborative teams that bring all of the capabilities and expertise that we could possibly bring to bear together, pulling in the same direction and truly believing in that outcome. And I think if you add to that the financial resources and the talented students and trainees that we also have access to through our great institutions, it really creates an immense amount of inertia and momentum towards this goal. And even though we've just started the seed projects for this collaboration, they're already bearing fruit, and I'm just incredibly excited to see where we're able to go in the next few years and beyond. So,
I guess I'll go next. And I agree with everything that's been said. And I'll add one other additional component. It's been fun. So it's been a pleasure working. I mean, for me, learning transplant. I mean, I dabble in it, but learning that. So it's a learning progress for me, and it helps to focus. I mean, as an engineer, we need to solve a relevant problem. So we need the end user there to help us walk along the way. So it's a definite partnership, and it's a lot of fun. So I'll leave it at that. And Tanya, I'll turn it over to you.
Thank you. It's kind of hard to go last when everyone said everything. I think Burchin answered for me for sure. But absolutely, Phil, it's been really great. I think selfishly, the thing that I love most about this is being able to tell, my patients or family members and friends that have kidney failure that we're out there trying to make a difference. One, we're trying to stop people from progressing to end-stage kidney failure. Then we're trying to increase the number of organs that are available in the pool. And then after transplant, we want to make sure you can keep that precious organ that you received. It's so fulfilling to be able to say that you're out there, you're working with individuals at CMU, you're able to sweet talk people, Adam, into starting to work on kidneys. And then I get to work with Keith Cook and Charlie Wren, and Phil, you've been mentoring us with the EV therapeutics. And I think that that's really what's been very satisfying beyond my day-to-day work, whether it be in research for chronic kidney disease, my clinical care, and also working with you all in the engineering fields. So thanks.
Thank you to all of you. I want to thank you, Adam, Burchin, Latanya, and Phil, for joining us for this discussion. I also want to thank everyone who's joined us here today. And I want to invite everyone out there who attended this to help CMU and the Mayo Clinic to do this transformational work that we discussed here today. And you can help us whether by volunteering to work with us, through philanthropic support, or simply just by talking to other people, spreading the word about the pioneer work that's being done at CMU and Mayo Clinic. So thank you again for joining us and taking part in this CMU community event. I wish everyone a good evening.